My name is Dean Brown. I'm an analytical scientist with Agilent Technologies. I'd like to introduce to you today the Cary60 UV-Vis dual beam spectrophotometer. Its design is simple yet efficient based on technology that has been proven over time and can offer many unique features to solve your analytical problems. I would like to discuss a few fundamental UV-Vis applications and show you how the instrument was designed to overcome those challenges that you face running each one of those applications. The ability to read small sample volumes is necessary in many applications, but particularly in biochemistry, where one has to look at small sample volumes and determine its purity as well as its concentration. This application requires an instrument that can read small sample volumes yet maintain the sensitivity of the measurement. I would now like to demonstrate how the Cary60 maintains the energy throughput regardless of the type of microcell or regular cell you use. Now, we're all familiar with the standard cuvette. It requires 3 mL of fluid to fill it. So now we're going to put this into the instrument as our baseline and see how much energy gets through a standard cuvette, and then we'll reference that to our other small micro cuvettes. So first I can take off the sample compartment lid because the Cary60 can measure with the sample compartment lid off. It has a technique or a feature called room light immunity, and that allows us to do enzyme kinetics reactions, quantitative analysis with the sample compartment open. So I will insert the cuvette now, and we'll take a measurement and see the energy. The second cuvette that we'd like to look at is called a macro cell. Notice it has thicker walls, and that of course takes up volume, but it still keeps the sample at the center of the cuvette. So this allows us to use a reduced volume as well. So now we're going to take and read that one, and this usually requires only about 200 microliters of sample. So I'm now going to place it into the sample compartment, and we'll repeat the measurement. The third cell we'll look at is called a biocell. It has a very small window. It's reduced in volume to about 30 to 40 microliters. And again, we'll measure this one. The fourth cell type we're going to measure, it's called a tray cell, that measures 1 microliter sample and can determine both the concentration and the purity of the nucleic acid, if that's what you're measuring. So we'll place it in the sample compartment and see how much energy we can get through with this arrangement. So as you can see from the experiment, that the first 3 transmitted the same amount of energy. In fact, all the energy that was coming from the instrument. The fourth cell, the tray cell, had a reduction, but when you consider that you were only looking at 1 microliter of sample, you would expect a little drop off, but you still have much higher signal to noise than you would with a standard designed instrument. The advantage of using the tray cell as opposed to a dedicated small volume spectrophotometer is that you can take the tray cell out and use your other standard cells for whatever application that you need to do. The design feature of the Cary60 that allows us to get this energy throughput is from a feature called focus beam optics. What we do is that we focus our beam down to be 1.5 millimeters by 1 millimeter. So all the energy from the instrument is focused down into that point, and that's why we can get through these small volume cells and maintain our signal to noise. Fixed point enzyme reactions require 2 things from an instrument. First of all, it has to be stable. The second thing is you have to have temperature control, because the rate of the reaction is dependent on the temperature. The instrument will record the change of absorbance with time. The Cary60 is a dual beam instrument. That means that the source is referenced so that you have stability. Typically, a dual beam instrument has 4 times less drift than a single beam instrument, and this is imperative for an enzyme measurement. We have a video that will actually show the ratioing system within the Cary60. Another point that's extremely important is that you have to have a fast collection rate. Now, of course, this depends on the rate of a reaction. A fast collection rate is imperative in enzyme kinetics. Of course, it's dependent on the rate of the reaction. But if you have a very fast reaction, you need to collect enough data points to determine or describe the reaction rate. Now, that's one of the outstanding points of a Cary60, in that its sample collection rate, or data point collection rate, is 80 points per second. Another problem that you run into in running an experiment is sometimes you need to extend it. You thought that the reaction would take maybe, let's say, 5 minutes, but now you realize that it's not going to go to completion. In the software, it accommodates this feature that it will allow you to extend the collection rate without corrupting the data for your enzyme reaction. One of the features of the Cary60 is what we call room light immunity. In one of our other modules, as well as this one, you will see that you can operate with the sample compartment completely open. So what does this allow me to do? Well, it allows me to add reagent, substrate, or enzyme during the course of the reaction so that you don't have to pause or stop the experiment. The xenon flash lamp also prevents photo-enhanced reactions going on in your enzyme system. Since the pulse lamp is on only for a short time, and the measurement is only made during that time, you do not get the spurious data from these enzyme-enhanced reactions as you may get with a continuum lamp. An alternate way of running a lot of samples is to use a fiber optic probe. It can be remotely triggered while you're running the sample, but the most important thing is that you have a defined path length. And of course, that's important for quantitative analysis. And depending on the samples that you have, different tips can be added to be compatible with whatever solvent system you're using. So now we're going to construct a calibration curve to show how easy it is. So I place the probe inside the vessel. I have to make sure that I cover the full path length, which I do. Now all you have to do is just click the trigger and the measurement is made. Now we come back and we run the second low standard. And again, press the button and take a measurement. So now we'll measure our middle standard. Again, we'll put the probe into the solution, press the trigger, and now the measurement's made. And now our final high concentration standard. So in a very short period of time, we were able to construct a calibration curve, and now I can measure all of my unknowns. And as you can see, a very large number of samples can be measured in a very short period of time. Now most people say you can measure powders, a solid sample, on a UV-Vis Spectrophotometer. Well, you need a special accessory, and that's called a remote DRA, or diffuse reflectance accessory. And what we're going to demonstrate today is we have some color tiles. We're going to measure those, and then we're going to compare them to the standard to show you that we can, in fact, actually measure the color of the system. And one other nice thing about this system is it also includes a video camera. So you can take a picture of what you're analyzing, and then you have the spectrum to identify what it is, and you have the complete answer. This is the diffuse reflectance accessory. It hooks up to the optical system of the Cary60. This is the alignment bracket, which we set the detector on. So we place this over the sample. The hole indicates where we are on the sample. And once we've done that, we simply put the diffuse reflectance accessory onto the bottom bracket. This is the detector, and this is the camera that will take a picture of it. So we're going to do this on 3 color slides, and we're going to see our results. So first we place the base plate on the sample. We verify that we're on top of the sample, and then we place the diffuse reflectance accessory on top of the base plate. Now we can take a measurement of the spectra of that particular tile, or we can take a picture. And the picture verifies where we are on the tile, so we can say that this is the color coming from that particular sample. So when would people use diffuse reflectance of a solid or of a powder? Well, for product uniformity, sometimes for counterfeiting. I see it, the colors are from one manufacturer to another, or from the counterfeiter to the manufacturer, or in art conservation studies where they're trying to make sure that this is in fact an old painting, because you can determine that by the pigments that have been used in the paint to see whether it's a new paint, old paint, or whatever. I would like to thank all of you for taking the time to view this demonstration of the Agilent Cary60 UV-Vis Spectrophotometer. I hope it was informative, and I hope it may help you in your future laboratory work and resolving some of your analytical barriers.